Today we're going to be talking about the role of the microbiome, the hot topic of the microbiome, and how we think it may be used as an alternative tool to improve the disease response of pigs using a PRRS PCV2 co-infection model. So let's start out with the basics. The microbiome is a term used to describe all of the microorganisms that literally share our body space. So this includes commensal, symbiotic, and pathogenic microorganisms. And the term was first coined in 2001, and since that time we've really learned a lot about the microbiome, and most of these microorganisms actually have a mutualistic relationship with the host, meaning that the host provides benefits to the microorganisms and the microorganisms provide benefits to us. We've also expanded the term since it was first used to include things like the soil microbiome and how can we utilize the soil microbiome to improve the growth of plants. So the microbiome, like we talked about, is a hot topic. It's an emerging area of study, and this is true for both the human side and the swine side. You can see for the number of publications has really increased over the last decade, but primarily within the last 3 to 4 years. So there's been just over 25,000 publications on the human side, a little bit further behind on the swine side with just over 500. But we're learning more and more with more projects coming about on the swine side as well as on the human side. So for the human microbiome studies, the emergence of this area has really been driven by the NIH Human Microbiome Project. And that was an allocation of $150 million to look at the differences in the microbiomes between healthy humans and those with certain diseases. We expect that the microbiome research will continue to emerge because of the recent release of the National Microbiome Initiative by the White House, where over $500 million has been allocated to understand how the microbiome is across different ecosystems. So there are several sites of microbial colonization in both humans and swine. We first think about the oral and nasal microbiome. The oral microbiome has primarily been researched with dental diseases and various other oral conditions. The nasal microbiome, interestingly, has been found to have various associations with development of respiratory disease. We also, there's been several studies in the urogenital microbiome, primarily in humans, looking at vaginosis or transmission of sexually transmitted diseases. We also know that there's a normal lung microbiome, so low levels of bacteria live in our lungs at a baseline level. And what's primarily been looked at for the lung microbiome is secondary infections due to HIV, also various pneumonia conditions, and diseases that are considered non-infectious like chronic obstructive pulmonary disease. In addition, the skin microbiome has been looked at various dermatological disorders such as psoriasis. However, with that in mind, the majority of the microorganisms that live within us and on us live in the gastrointestinal tract. So these microorganisms are estimated to weigh about 3 pounds in the adult human, and they outnumber our cells about 10 to 1. These are diverse. They include bacteria, viruses, fungi, protozoa, and archaeobacteria. And we'll be talking about primarily the gastrointestinal microbiota. So there's several roles of the microbiome, both intragastric and intragastrointestinal, as well as outside the gastrointestinal tract. So we know that the microbiome has a large role in metabolism, and there's regulatory molecules that are absorbed through the gastrointestinal tract due to the microbiota that circulate throughout our entire system. They also play a major role in immunity, which is obviously a systemic thing. So both intra and outside the gastrointestinal tract, these microorganisms play an essential role in our health. So first, the microbiome plays a role in absorption, metabolism, and storage of calories. This is particularly true for those types of calories or nutrients that we cannot degrade on our own, such as complex carbohydrates and fiber. We also know that the microbiome plays an important role as a protective barrier. So this is both through competition of nutrients and just through sheer volume of the normal flora, impeding those pathogens from gaining access to the gastrointestinal tract. And finally, we know that the microbiome plays a major role in both the development and regulation of immunity. We know that it plays such a major role in the development of immunity when we look at germ-free mice compared to those mice with normal microbiota. The immune systems are underdeveloped in those mice without normal microbiota. There's less antibody production, there's less cytokine expression. So we know that the microbiota and that initial exposure to microorganisms is really important in stimulating the immune system in the neonate and then continuing to regulate the immune response to pathogenic and non-pathogenic organisms throughout the life of the animal and the human. So in the swine, within the first weeks of its life, we know that there are several factors that impact microbiome development. And so these initial factors have a lot to do with the sow. The piglet has no microbial colonization when it's still in the uterus. And as the piglet exits during delivery through the vaginal canal, as you can imagine, the vaginal microbiome of the sow plays a major role in that initial ingestion of microorganisms during delivery. We also know that both the diet and the gastrointestinal microbiome of the sow play a major role in microbial colonization of the neonate. We know that if we modulate the microbiome of the sow in the gastrointestinal tract, that will in fact modulate the microbiome of all the piglets. Due to exposure to feces, the skin microbiome, everything on the sow has a lot to do with the initial colonization of the piglets. We also know that environment plays a major role in the microbial colonization of these piglets. So there's been several studies looking at outdoor housing versus indoor housing versus isolator-reared housing. And as you can imagine, the outdoor exposed pigs are exposed to a lot more microbes early on in life than an indoor or an isolator-reared piglet. We also know that the diet of the piglet is essential for microbial colonization, and that the transition to solid feed for the piglet is one of the major determinants of microbial colonization, and it has a lot to do with carbohydrate and fiber source. We also know that stressful events at processing, such as needle teeth clipping and tail docking, of course injectable antibiotics is a little more inherent, that impacts the microbiome as well. So stressful events as well as antibiotic administration can impact the microbiota for several weeks. And finally, enteric pathogen exposure, as well as any pathogen exposure, will cause shifts or changes in the microbiome. In particular, PEDV, we know, causes a dysbiosis and a reduction in microbiome diversity. In addition, in-feed antibiotic growth promoters, we know that one of the mechanisms that works to enhance growth is through shifting the microbiome populations towards a microbiota that have an increased capacity to harvest energy. So how we can utilize the microbiome to enhance growth performance without the use of antibiotics is really the question. And understanding that there's all of these factors that impact microbiome development, as we understand more and more about the beneficial characteristics of the microbiota, we can see that there are several areas where intervention may allow us to enhance and benefit pig performance and response to disease in the early life of the pig. So the microbiome's role in health and disease is very complex. It's not well understood, and I loved a lecture this morning by Jim Lowe when he said, we know what, but not why, because that applies a lot to the microbiome. We know several changes are associated with certain diseases in humans and swine, but we don't necessarily know the exact mechanism. And as you can imagine, with the diversity and the thousands of microorganisms, these relationships are very complex and complicated. However, several publications have shown that microbiome diversity or composition play a role in both disease susceptibility and response to pathogenic infection. For example, the inherent relationship that's sort of easy to understand is how the gastrointestinal microbiota would impact diseases of the digestive system. So we know that Clostridium difficile overgrowth is associated with a reduction in microbiome diversity. And in fact, one of the leading treatments right now for C. diff overgrowth is to have a fecal microbial transplant, where you're taking feces from a healthy adult and you're transplanting that into someone with overgrowth of C. diff. That has a higher success rate of eliminating the pathogenic overgrowth than antibiotics targeted at C. diff alone, which is interesting to think about how microbiome diversity is really important to several human diseases, and we'll see a similar effect on swine. Obesity is also associated with shifts in the microbiome, so we know that obese humans and mice typically have increased Firmicutes phylum bacteria and a reduction in the Bacteroides phylum bacteria. And in fact, obesity is so closely linked to the microbiota that if you take the microbiota from an obese individual and put it into a germ-free mouse, that mouse will actually become obese. So it's one of the few diseases where they've shown transmissibility through the microbiome. We also know that the gastrointestinal microbiome plays a role in several neurologic diseases. So the development of autism is associated with an increase in Clostridium species, and chronic fatigue syndrome, similar again, is this common theme of increase or decrease in microbiome diversity being associated with several diseases. And finally, we know that the gastrointestinal microbiota plays a role in several lung infections and non-infectious disease, like asthma and allergies. We also know that pneumococcal pneumonia in a mouse model has increased bacterial load in the lung, increase in the pathogenesis of the lung lesions when there's a reduction in microbiome diversity. And similarly, if you have a Helicobacter species and infected with TB, you have more inflammation in the gastrointestinal tract that leads to higher pathogenesis of TB in an infection model. So it's interesting to think about these relationships. So what we know from the human side and some work in swine is that the microbiome impacts both weight gain and immunity. So what we wanted to do through our research was to look at how does the microbiome impact PRRS PCV2 co-infection. We know that both viruses cause systemic infections with primary lung pathology. The two viruses together enhance disease as opposed to a singular infection. And we also know that both viruses modulate the immune system. So we thought there may be a role here for how the disease outcome occurs in pigs post-infection. So our first question was really, are there any microbiome characteristics that are associated with outcome following PRRS PCV2 co-infection? To answer this question, we had 95 nursery-age pigs that were group housed at Kansas State University. All pigs were challenged with PRRS PCV2 and followed for 70 days post-infection. At 70 days, 20 pigs were selected for having either the best or worst clinical outcome, and fecal samples were collected at that time for microbial analysis. We selected these pigs by first ranking average daily gain from highest to lowest and clinical disease duration from shortest to longest. 10 of the best clinical outcome pigs were selected due to having highest average daily gain and a complete lack of overt clinical disease. The worst clinical outcome pigs were selected due to having the lowest average daily gain and 10 or more days of moderate to severe clinical disease. We confirmed the divergence of these two groups in weight gain over the course of the post-infection period. And what you can see is, let's see if I can get this to work here. So the differences in weight gain really start at 7 days post-infection. So within 1 week post-infection, the weight gain between the two groups had diverged significantly. And then over the course of the 70-day co-infection period, they continued to diverge and showed that the best performing group had a significantly higher average daily gain throughout the post-infection period compared to the worst performing group. We also confirmed that the time course of clinical disease during the 70 days post-infection was consistent in the worst clinical outcome group shown here with the entire population. And what's really interesting in our PRRS-PCV2 co-infection model is that it's really a biphasic disease response initially associated with PRRS virus replication and secondarily associated with PCVAD. The clinical signs in these pigs were diverse. Again, another advantage, if you will, of using a co-infection model. We see lots of different clinical signs, and so we can hope that any microbiome characteristics that we can pick up would be broadly beneficial for these multisystemic diseases. However, all the pigs showed clinical signs of respiratory disease, dyspnea and rhinorrhea. We also wanted to look at other pathogenic factors of the co-infection model, such as lung lesions and PRRS virus replication, to again see if there were associations with the microbiome. For lung lesions, we score the lung from 0 to 4, 0 being normal lung shown here, 4 being severe interstitial pneumonia. And what you can see is the worst clinical outcome group had significantly higher microscopic lung lesions when compared to the best clinical outcome group, again confirming that severity of interstitial pneumonia plays a major role in clinical outcome. We also can see that the PRRS virus replication over time was higher in the worst clinical outcome group shown here on day 28 post-infection. When we took the total virus replication by calculating the area under the curve, we again found that the worst clinical outcome group had significantly higher total virus replication when compared to the best clinical outcome group, again confirming that pathogen load plays a major role in clinical outcome, but also enabling us to associate any microbiome characteristics with lower lung lesions and lower virus replication. To assess the microbiome, we use the Lawrence-Livermore microbial detection array, which has the ability to detect over 8,000 species of microbes. It's designed to detect any species of microbes that's been sequenced. We first looked at microbiome diversity between the two groups. And what we found was the best clinical outcome group had significantly higher microbiome diversity as calculated by the number of families when compared to the worst clinical outcome group. Again, similar to the human diseases in that microbiome diversity is beneficial to clinical outcome. We also looked at the microbiome composition and found several bacterial and viral species. However, most of the bacterial species are found at similar rates between the two groups. When we looked at E. coli, however, non-pathogenic E. coli, we found that this was found at a significantly higher rate in the best clinical outcome group as opposed to the worst clinical outcome group. And when we took a step back and looked at the overall Proteobacteria phylum, which typically only makes up 2 or 3% of the overall bacteria in the microbiome, again, mostly Firmicutes and Bacteroides, we found that proteobacteria were only found in the best clinical outcome group, indicating that the members of this phylum may be beneficial to clinical outcome. We also found several viruses in the feces of these pigs. However, none of these viruses were found at significantly different rates between the two groups. So to summarize, we found that both increased microbiome diversity and increased non-pathogenic E. coli were associated with improved clinical outcome 70 days after PRRS-PCV2 co-infection. So we found this is very exciting because we didn't initially know if we would find differences in these clinical outcome groups after co-infection. But it also led us to the question of, did the microbiome characteristics change during the course of co-infection, or were there microbiome characteristics that were present prior to co-infection that helped predict or predispose outcome? So this led us to the question, are there microbiome characteristics that predispose or predict outcome to PRRS-PCV2 co-infection that would help us modulate the microbiome prior to exposure to help improve clinical outcome? We answered this question by using 50 nursery-age pigs that were again group housed at the Kansas State University. All pigs were challenged with PRRS and PCV2 and followed for 42 days post-infection. At the conclusion of the trial, 20 pigs were selected for either having high or low growth rates. However, we had collected fecal samples from all 50 pigs because we're unable to predict outcome at day 0. So we were able to go back for those 20 pigs that we selected from the high and low growth rate groups and analyze what microbiome characteristics predicted or predisposed the differences in outcome. We selected these pigs by first ranking average daily gain from highest to lowest. We excluded all the pigs with clinical disease from this study. This was for several reasons. One is that pigs that showed clinical signs during these co-infection trials were typically handled by a veterinarian, could have been undergone stress due to the handling. They may or may not have been treated with veterinary drugs. And we wanted to take out the clinical disease from this experiment. So basically what we're looking at here is what microbiome characteristics shape the growth rates after co-infection in all subclinical pigs, so no pigs showed clinical signs of disease. So we selected pigs from the high growth rate group due to having high average daily gain, and again, a complete lack of overt clinical disease. Low growth rate pigs were selected due to having the lowest average daily gain, and again, no overt clinical disease. We confirmed the divergence in weight gain between the two groups over the course of the 42 days post-infection. But as you can see, the divergence between the two groups is more subtle in this experiment, but it does become significant at both 35 and 42 days post-infection. In addition, the high growth rate group had an overall significant increase in average daily gain during the co-infection period. When we looked at lung lesions and PRRS viremia, we first found, again, a trend towards the low growth rate pigs having higher lung lesions, which was initially surprising. Again, no pigs showed clinical signs of disease. But as you can see, in subclinical pigs, the level of interstitial pneumonia is still affecting their growth rate and outcome. We also found that the PRRS virus replication was higher in the low growth rate pigs. We found that on both days 4 and 14 post-infection, and when we looked at the overall virus replication or area under the curve, we again found a significant increase in the low growth rate pigs post-infection. Again, interesting because no pig showed clinical signs of disease and showing how much of a production disease this is by reducing the growth rate. When we looked at microbiome diversity, we similarly to the first experiment looked at family diversity first. And what we found was that the high growth rate pigs had a significantly higher family diversity when compared to the low growth rate pigs. We took this analysis a step further and looked at species diversity and found a similar trend. A significantly increased microbiome diversity in the high growth rate pigs. Again, this is at day 0. So these pigs were identical weights at day 0. However, they had these significant differences in microbiome diversity prior to co-infection that likely was associated with the growth rate post-infection, showing again microbiome diversity is beneficial to outcome. Yeah, okay. We also looked at the composition of the microbiome and found 29 families. However, the percent that these families were detected in the two groups were not significant throughout all of the 29 families. So we had to take this another step further as well, and what we looked at was the number of species within each family. So you can see like lactobacillus, which is a very common bacterium, had the most species of any of the families we detected. But these species were not different between the two groups. There were 3 families that stood out as different when we looked at the number of species within family. The first one was Methanobacteriaceae, and this was an interesting one because this was the only family that we found where the high growth rate pigs had significantly fewer families. So typically in what we've talked about so far, the better outcome pigs have either more diversity or more of the bacterium. This one was different in that Methanobacterium may be detrimental to growth rate after co-infection. We also found that the Ruminococcus and Streptococcus families had significantly higher numbers of family in the high growth rate pigs, so similar to the first experiment, indicating that those 2 families may be beneficial to clinical outcome. So to summarize, we found several characteristics of the microbiome on day 0 prior to co-infection that were associated with high and low growth rates after co-infection. So we found several characteristics of both the microbiome on the top that were associated with improved phenotypes of disease response. So increase in E. coli, a reduction in methanobacteria, an increase in Streptococcus, specifically Streptococcus equi, and an increase in Ruminococcus species. And they were all associated with these various disease phenotypes, which is interesting when you look at the literature because you can find similar patterns. For example, methanobacteria is associated in humans negatively with body mass index. And so when you look at studies in humans, people who are anorexic or lean have higher methanobacteria than, say, an obese human. So some of these similar characteristics have been found in humans, and it's interesting to think about how they may correlate to improving pig performance in swine production. But what was really interesting is that this increase in microbiome diversity was consistent between both experiments. So both at day 0 and day 70 post-infection, microbiome diversity was beneficial to both reducing clinical signs and improving growth rate. And so the question is, how do we improve microbiome diversity or increase it when we have several practices to reduce pathogen exposure and biosecurity? So really, in both humans and pigs, I think the question is increasing microbiome diversity without increasing risk of pathogen exposure to improve response to disease. And ultimately, how can we use the microbiome to our advantage to improve pig health, to improve the response to various diseases, and overall create a more robust immune response and improved growth performance in the pig?